All right, good evening. This is IRP recording number 3 for the GMO project that Manny and I are doing. So first off, what our group experimental plans were this week was we were planning on running our PCR for our original 4 samples that we had of the corn husk, the tortilla, and the Doritos. And we're going to run those with primers that were specific for the corn mosaic or cauliflower mosaic virus, which is the GMO sequence that we were looking for or trying to identify, and then the other primers were just the corn DNA primers in general. The reason why we wanted to do both and we were doing them separately is so that we could identify just the corn DNA, make sure that we had some in the first place, and then we wanted to do the other ones separately just to try and isolate just that sequence. The previous group had done both of them at the same time, and when they got their bands were kind of they weren't very clear, and so we wanted to run them separately to see if we could get a better result. Along with that, we were going to isolate more DNA from different samples of like Frosted Flakes or corn nuts or Funyuns, and even some Fritos, just different corn products that we went and got this week that we wanted to isolate more DNA from. And then we're also going to isolate DNA from our original samples again so we could try and replicate this process that we've been going through. So what we were able to accomplish is we were able to isolate more DNA, but we haven't been able to do the nano drop yet. We were going to today, but we ran out of time. So we're going to go in tomorrow morning and isolate it, or we'll go in on Monday again like we did this week and use the nano drop to find out that we've got DNA. Along with that, we also ran those PCRs, and we ran a PCR on Monday, and we ran it on the gel on Tuesday, and unfortunately we didn't have any bands that show up. The only bands that showed up were the ladder molecular marker, and so we went back, did some troubleshooting, realized that we needed to add more DNA because we had done the amount that was in the lab when we did this. So we added in 500 nanograms of DNA, or as much as we could, in our samples. Our DNA was kind of low. So we did that, and then we ran another PCR and ran it on a gel, and we had some bands for our tortilla sample and for our Doritos sample, just for the corn DNA, though. Nothing for the GMO sequence that we were looking for yet. Along with that, we isolated all that DNA. And then for my individual contribution, I stayed late on, I think it was Tuesday. We were isolating, or not isolating DNA, we were preparing our PCR samples, and I had to stay late because we had started them, and Manny had to take off or something, so I stayed late so we could finish them. And then I even came back later because the PCRs were being used at the time, so I came back like an hour later so that I could throw our PCR in, so that would be ready for us on Wednesday, which is when we ran our second gel. Other than that, I also created an Excel sheet that we were able to use to do quick calculations for our samples based on their concentration of DNA that we got from the nano drop, and that we could then use to just calculate how many microliters of that we needed to add into our PCR tubes. Challenges we faced this week: we were running out of reagents. The plant genome kit that we were using didn't have very many reagents left in it from whoever used it last. So we were running out of things like our RNase A, and we were also running out of some buffers. But things that we were able to do to face those challenges is I talked with Sister Walker and concluded that we could use some buffers from other genomic DNA isolation kits. And so I talked with Manny, and we decided to use those. And then we decided that for one of our samples, the tortilla, which we did get protein bands from, we were going to do completely with the genomic DNA kit to see if we could get better results isolating DNA from that versus doing the plant genomic DNA isolation kit. And so those are the things that we worked on, and we're going to be going in, like I said, tomorrow and Monday to use the nano drop and to run a second PCR. So hopefully we'll be able to have some more results, especially with this being our third PCR. We'll hopefully have the process down to be able to get some better bands and hopefully find some GMO sequences.